Whoa. So where do I start? So thank you. Thank you, Patrick, our president of the Max Planck Society, for these kind words. Thank you, Auguste, who's actually a colleague at the Max Planck Institute of Biological Intelligence. Thank you, Michael and Michael, for allowing us to host this event in this venues and beautiful place and in the Nymphenburg Park. Thank you to the donors for supporting this event. And also thank you to the EMBL Alumni Association, Ian, to Matthias, for the honor to give this lecture in memory and really in celebration of Fotis Kafatos. And also thanks to the friends and colleagues. Also my lab is here, who are here, members of the public, who are here in the audience or connected virtually. So what I would like to do today is discuss with you work we've been doing to answer a problem that has really been captivating us. And that is, how is the life and death of the mRNA molecules in our cells controlled? And to set the stage, I need to go to the fundamental principle of life. And that is the sequential flow of information that from DNA, which contains the genetic instructions, is copied, transcribed into RNA. And from there, it is translated into proteins. And as Patrick said, proteins perform the bulk of the work in our cells. They catalyze chemical reactions. They form the structure of our cells. They signal. They function as signaling molecule with the environment, transport, etc. Now, today I will focus on messenger RNA, but just would like to tell you there are many kinds of RNA. RNA is a wonderful molecule, and they do a lot of work, a lot of functions, including catalyzing and supporting and signaling. So there is much more to RNA than what I will discuss with you today. So this is synthesis, but there is also degradation, as Patrick mentioned. Now, DNA is a permanent feature of our cells. It is very stable. In fact, as you know, it can even be extracted from the bones of Neanderthals, as you know from Svante Pääbo's work in the Max Planck, that have been in a cave for 40,000 years. Now, RNA and proteins are not permanent. They are transient molecules. They are made, they do their job, and they are degraded. And it's actually not just the synthesis of RNA and proteins, but it's also the degradation that regulates, it's the balance between synthesis and degradation that regulates the amount of these molecules in our cells. Now, degradation is not just used to turn over these molecules. Degradation is also used in case there is a problem. And the problem may come from errors in these processes, which are actually, by the way, made by proteins, transcription and translation, as you'll see later, or it comes directly from mutations in the DNA. And when I was thinking how to, kind of convey this problem, it's a little bit what happens in an office, okay, where you have this shredding machine that which are used to fragment documents that are either not needed anymore. So these man-made machines, of course, that are documents that are no longer needed, or they contain potentially hazardous information if they would leak out. And similarly, in the cell, somewhat similarly, the cell has molecular machines, and these molecular machines break down RNA molecules that are either no longer needed or that are defective. And you may imagine, probably from your own office, maybe not, what happens if one doesn't get rid of things you don't need, of documents you don't need. The cells obviously would be overwhelmed by these molecules that are not useful and not needed. And so the questions that we've been interested in the lab is, how do this molecular machine recognize and eliminate RNAs? How do they recognize which RNAs to degrade, which one is defective or superfluous? And how, which parts of these machines are compromised in disease mutants? I'm not going to talk about this part today. Now, there are several machines that are involved in RNA degradation. And one that we have been working on for many years, as you will see in the next slide, is a machine called the exosome. Actually, there are several sorts of exosome complexes. They are usually depicted here as this kind of Pac-Man molecule ready to attack the 3' end of the RNA. They recognize the 3' one end of the RNA, and they degrade it. One nucleotide, they cut it out, one nucleotide at a time. And this was actually a machine that was discovered by David Tollervey when he was at EMBL. Now, this machine exists in different types, depending on the context, although the heart of the complex of the molecules are the same. And it acts on all types, virtually all types of RNAs. It's actually a very ancient machine. Exosome-like complexes are present in all domains of life. There are exosome-like complexes from bacteria and all the way to humans. Now, my lab started to work on this, imagine, 20 years ago. Okay, so we started when we were at EMBL, and we decided to tackle the problem starting from a simple organism, from an archaea organism, with the idea that we would start learning from the simplest system and then progressively move to the more complex system. And when in 2007, then we moved to the Max Planck Society, then we transitioned from the archaea to the eukaryotic system, again starting from the simple eukaryotes, from yeast, to then study these machineries in human cells. And what we are interested in studying is the molecular mechanisms, the mechanisms with which these molecular machines work. And we want to see it at the atomic level to understand how it works. And studying, of course, the human complexes is, of course, important because there are many mutations, and again, I'll not discuss those in detail, but there are many mutations in subunits of this Pac-Man here, which are connected to disease. So how do we study a macromolecular machine at the atomic level? And it's a little bit, so if we want to understand how these machines work, we have to understand how they are built. So it's a little bit like reverse engineering, okay? You try to understand how a machine is working, like here, by taking it apart, seeing not only what the components are, but how the components come together. Now, the analogy, of course, has a problem when we go to the molecular machines, and the problem is just the sheer size. So the core complex, so we are talking just about the heart of the exosome complex, which is about 10 proteins. If you look at the maximum dimensions and the weight, now we measure those in angstrom and in Daltons. Now here is in meters and in grams. It's just unimaginable, just even to think about how small these things are. So how can we see them at atomic resolution? And the answer is using methods that come from physics. In the lab, we use X-rays and electrons, and so the technology comes from physics. The questions, of course, is a biological question, but what we want at the end is to interpret the results at the chemical level, and of course the relevancies for medicine. So I thought I'd show you some data. This is a movie that Christian made, and these are on many different structures. So here is the end, it's actually the cutting part of the complex. And what you're seeing here is the distribution of electrons, okay, in this molecule. And now what we do, we recognize features in this, and these are the building blocks of proteins. These are all the atoms over here, which fit in the electronic distribution. So we interpret the electronic distribution by placing these building blocks over here. And slowly and surely, we build the entire molecule. So we build the entire 3D architecture of this protein. Now, as you see here, there is some other density here that is unoccupied. This is actually RNA, which is now building up here. And the reason we could catch it is because we changed one atom in all these machineries. There's going to be thousands and thousands of atoms. We changed one oxygen to nitrogen, so we could kill the enzyme. Now, this doesn't work alone. It works as a complex of 10 proteins, as I told you, which are shown here. And what you will see appearing here is the RNA coming in, channeled in the machine. This is the threading side. This is the entrance, the feeding end. Now, this doesn't work on its own. Obviously, it works as part of a larger complex. Now, this is a cryo-EM, so now we are moving to electron microscopy, and it's a much larger complex. In this case, this machinery, the exosome, we managed to stall it as it's grabbing an RNA molecule. This is not messenger RNA, it's part of the ribosome, which also has to be matured. And from there, we can go and we can look at the active site. Now, this is the single nitrogen we changed. This normally would be an oxygen. And when there is an oxygen over here, the reaction will go. So this is yeast here with this little scheme over here. But actually, the architecture of the complex has been conserved through evolution. So this is the structure in yeast, which I'll just show you. This is the structure from human work from us and also from Chris Lima's lab at Sloan Kettering. And you can see there are some details that are different, but the overall structure is very similar. But as I told you in the slide before, this doesn't work alone. So this is like this part here, these 10 proteins are, I would say, like this part. It's the shredding unit. It has the feeding slot up here where the RNA will go in, and this is where it's going to be cut. And like in the man-made machine, you need something else. Okay? You need the hand that recognizes the paper you want to cut, you want to feed, and that pushes it through. Okay? And so this complex actually works with other factors and other complexes that can target the substrate and feed it through this entry here. And as you will see, there are also sensors that I will show you in a movie later. Now, there are many of these factors, okay? And there are many complexes. They are different. They are in nucleus and in the cytoplasm. These are parts of the cells. And what they have in common is this blue thing over here, this blue blob over here. This is an ATPase. So like with the hand, this is the hand, okay? And like the hand needs energy to put something through, and this is an ATPase, okay? It uses ATP as energy to get the substrates. There are all kinds of different complexes like this because we have all kinds of different RNA substrates in the cell. We have been working on all of them actually, but today I'm just going to focus on one, okay, which is this one here. And this complex functions where this machinery, the exosome, has a particular problem. And the problem arises when we synthesize messenger RNA. And so what happens, this is RNA polymerase II, which actually Patrick has worked for a long time. And the RNA polymerase II is the machinery that does the transcription. So you see here now the DNA, and you see here in the schematic representation the messenger RNA coming out. This is the transcript. Now, what we know is that polymerase goes through, the machinery goes through, copies the gene into the messenger RNA, and then the mRNA is cut, and it's packaged, and then it goes to whatever it needs to do. It's exported to the cytoplasm. But actually what happens, particularly in human cells, is that the polymerase stops. And then there is a premature termination. And this happens quite often, actually. And so what would happen is that here you have a prematurely terminated transcript. And the cell has to degrade them because you don't want to make shorter proteins. This would be the blueprint for a protein. And so there is a massive amount of these kind of transcripts that have to be recognized. And this is where this complex comes across. Now, there are many complexes here. There is the exosome, there is the hand complex that puts things in. This is a complex that binds one end of the mRNA as it comes out of the transcript. There is RNA polymerase II. And so what our group, and I should say this complex was discovered by Torben Heikensen's group in Aarhus. And so what structural biologists, like my group, Patrick's group, have been doing over the years is to collect snapshots of these complexes at very high resolution. And so this is the core of the next complex over here. It's actually a dimer. It's even more complicated. And this is actually the core that was done by cryo-EM. But it's not just the core. There are lots of flexible regions which are now here predicted by AlphaFold, which is an artificial intelligence-based program tool, which just gives us a hint of how these unstructured regions that we cannot see by X-rays or by cryo-EM because they're just flexible, how more or less they would be. And of course, we have RNase polymerase II structures from Patrick's lab. We have structures here, the cap binding complex of the proteins here by Stephen Cusack's lab, also EMBL structure of the exosome from my lab, and also Chris' lab. And so what we can do is to take these snapshots. It's like this famous horse in motion. You have these photographs. We can take these snapshots and try and connect them to just have an idea of what the process would be like. And so what I'm going to show you now is a molecular movie. It's an animation that we have done with the lab of Janet Ibaza, where we combined multiple structure determined experimentally with predictions by AlphaFold, with information from biochemical and functional data that gives you an idea of what we think the process is molecularly, and gives us an idea what we do not understand yet. And so let me start the movie. So here you'll see polymerase II is in gray, the DNA is going to be in blue in this case, mRNA is white. It's already been capped at the 5' end with the cap binding complex. So let me play the movie. And now at this point, the molecules are on the same scale because they are based on real structures. So as the mRNA comes out, there are other proteins that bind the cap structures. Stephen has worked on. There is this protein in green, there is this other molecule. When the premature termination event, somehow this complex that I told you about, the next complex, is recruited. And this complex, which is now shown here with the various different proteins, is going to interact with the cap, with the mRNA, thread the mRNA through the ATPase. This is the hand, recognizes the first sensor goes, the second sensor goes, and now this is threaded in the machine, where the RNA is going to be degraded one nucleotide at a time. And in this case, the movie is a little bit misleading, actually, because this reaction is super fast. So you wouldn't be able to see. This is just slow down. So this is how prematurely terminated transcripts are degraded. There are many questions that, that are still open about this. But there is even, I think, a bigger question. What happens with a real messenger RNA? It gets out, okay? How is this messenger RNA protected? And this begs the question, well, what is the structure of these messenger RNA particles that come out? Yeah, and we know they should be covered with proteins. This is a lot of effort from Matthias' lab to identify proteins that are bound to messenger RNAs and are covered. Now, so what Fabiano in the lab has done was to purify these particles, and this has been very challenging because these particles are very fragile. And so this thing here, just for the students, is 3 years of work, okay, to get to this, yeah? And with this, he has enough material. This is again from yeast. So again, like in the case of the exosome, we start with a simple organism so that we can learn and then move steadily our way up to the human, okay? So this is yeast. Now, this is essentially, these are all messenger RNAs in the nucleus of yeast. So the population is very heterogeneous. We cannot use cryo-EM and X-rays to look at the structure. And so we have to try and look at it with a heterogeneous population in a different way. So these are the kind of experiments we are doing these days, just for you to have a feeling. So this is a cryo-electron tomography of the particles. And what you see here, they're all different because there are all kinds of different mRNAs, mRNA particles, but they're very compacted. And once we see they're very compacted, then we have to try and understand what compacts these particles. They have these very long mRNAs, so there must be something. And these long chains are very negatively charged. They have to be able to collapse to get to these structures. So what happens? And so to peek into this, what we use is cross-linking mass spectrometry. We combine with artificial intelligence tools, with AlphaFold, and with biochemical assays, which I am now going to show you. This, for example, is a proximity mapping, which is mapped with AlphaFold, which predicts protein-protein interactions and protein structures. And the picture that we get from all this data is something like this. So there are these molecules that are very specific molecules, and they are like they're structured cores, and then they have these long unstructured regions. They are like the tentacles of this, what do you call it, the polypo, you know, the thing in octopus, thank you. Ooh, thank you. I'm in the Mansion Nature Museum. Okay, sorry, the tentacles of the octopus that you see here. And it's early days, but we thought, okay, how about we try again and put the information or what we think is happening in a movie, you know? And this is work in progress, so there are going to be part of these movies that are not correct, and I will tell you which ones at the end, but just for you to have an idea. So let's start again. We start again. RNA polymerase II, actually this is a nucleosome, which is on the DNA. This is probably Patrick's structure, actually. I don't look at the polymerase, Patrick, just look at the RNA over here, okay? Okay, it's capped. This is the RNA coming out. There is another complex that comes in, this yellow complex, the tau complex, and here are the octopus molecules. And what we think they do, they come in, and with the tentacles, they have positive charges that essentially helps to get the RNA close together with the tentacles, and then they have the structured core that then reinforce. That reinforce the interaction. So essentially, and I just skipped the last part of the movie, sorry, I should have said, so what's not correct in that movie? You have seen molecules flying in magically. They're not going to fly in magically. The octopus is going to be loaded, okay? It doesn't come in magically. You know, the nucleus is very crowded. And so we're looking how it's going to be loaded. We are looking now at how it is loaded. It probably requires the yellow big protein that was present in the previous slide, which also was flying in magically. It's not going to fly in magically. It's probably has some way to sense the elongating polymerase II. And so these are all aspects that we are looking at it now. And so the movie that I showed you, which is work in progress, over the years is going to change by work from the lab that's going to come out. So this is what we see. So this is what we think is happening in the particles, mRNAs, nascent mRNAs in the nucleus in yeast. But actually, all these proteins are either conserved in humans or they have similar proteins that we think do similar job in human. Of course, in human it's going to be more complex. And so the way we think about it, and actually I was talking with Yulia in the lab, and Yulia had a really clever idea. Well, it's actually origami. So if you think about this piece of paper, again, to this analogy that we have made before, that goes through the shredding machine, what the cell seems to do is actually to fold, like an origami, the mRNA. And as you can imagine, an origami is not going to go very happily through the shredder. And so it packages this in a compact manner, and in doing so, we think it helps transporting through to the cytoplasm and protecting it. Now, why should you care? Well, first of all, it's a fascinating problem. This is why you should care. This is why we care, obviously. But like many basic biological questions, they have direct implications on our life. And I think there is no better examples in this case with what happened during the COVID vaccines and during the COVID pandemic and the mRNA vaccines. And these vaccines, I would argue, you know, we face a similar problem. So what they do here, this is this very clever way where the mRNAs are modified and they are encapsulated in nanoparticles, and they're kind of also packaged. And when these RNAs get in our cells, well, there is a problem. And the problem is that RNAs, I told you, is a very fragile molecule. Actually, this is why, as you know, these mRNAs, these vaccines, they have to be made in sterile conditions because you have RNAs on your fingertips. Okay, if you touch it, you're going to kill it. You're going to degrade it. And they have to be transported at very low temperatures. Once they enter your cells, they have a limited lifespan because they are degraded. And so the way around it is to try and boost the amount of mRNAs in these vaccine particles to try and get them to work as, you know, with sufficient time lengths. Now, of course, our cells are not at ultra-cold temperatures. And I do think that if we understand the principles, now, of course, we do not work on vaccines in the lab. We are working on the basic problem. But I think if we do understand how nature has solved the problem and how packaged and how they are protected, I do think that this is going to help the next generation, as we heard, of mRNA therapeutics, as we heard from what Patrick said, that I think is going to revolutionize our life and our health. And with this, I'm going to thank the people who have done the work. Actually, my lab is here. Part of my lab is here. Here we were in a lab retreat on beautiful Lake Stamberg. As you see here is a very international lab, like EMBL, like the Max Planck Society in general. And I would like to thank the Max Planck facilities, without which we couldn't have done this work. Danny Nediakov is a group leader in our institute. It's been great to, is an expert on mRNA, so it's been great to discuss. Janet and Margot, they are from the animation lab in Utah. It's great to, just to get the atomic level information to the kind of movies I showed you, because as soon as you try to make the movie, you understand what you do not understand. You know what you do not understand, and that's the question that you need to go after. And Torben, with whom we've been collaborating, we have been blessed with great funding from these foundations, from the Max Planck Society, thank you, from EMBL beforehand, from the DFG, thank you, and from the ERC. And I really would like to thank Fotis. Now, Fotis, I was EMBL Director General when I joined the lab, so Ian actually hired me, so I'm really, really grateful to him, and I'm really, really grateful. He was the ERC founding president. And for us, the ERC has been great because it really pushes you to go for the big questions. I mean, the Max Planck as well. Sorry, Patrick, Max Planck as well. But it keeps you on your toes. Max Planck as well, Max Planck as well. And with this one, I really would like to thank. These are the people, and these are really the people who drive all the projects I showed you. They are the ones behind, and they are the ones who are fascinated, and they are there to work and try and solve the problem. And with this, I would like to thank you for your attention.
Dear Elena, what a super inspiring lecture it was. Thank you so much for taking us on a journey to the absolutely fascinating world of RNA, and for sharing and explaining some of the very interesting insights of your research.
Thank you.
My name is Agnes. I'm coming from EMBL's Science Engagement department, where our aim is to bring cutting-edge science to your classrooms, to your schools, and where we want to have conversations with all of you and create opportunities for such conversations where we can discuss some of the most exciting topics in life sciences research. And today is a fantastic opportunity to do so. Thanks to Elena. I'm going to be handing over to all of you very shortly for questions. In the auditorium, my colleagues will be walking around with a microphone. And if you have a question, please raise your hand and we will come to you in a minute. We will be gathering questions from the online viewers as well. So please do post your questions into the Q&A section of Zoom, and we will look into those and address, of course, those as well. Elena, just to start off the conversation, may I ask you what inspired you to become a researcher?
Oh, I didn't expect this question.
Was there a special moment?
No, it's okay. I have a very different background. I come from a small town in Italy. My family is not, they are not scientists. It's a very different background. I was fascinated by chemistry because I had a very good teacher. I was also kind of good, it helps if you're good at it also too. And then this led into biochemistry and the step after the next. I never had a big career or life plan. Yeah, one step after that step. And for me, it was like a passion that grew. And I feel very grateful that I do something I love every day. So I think I knew how to, I wanted to do this after my postdoc, I guess.
That's lovely, and I'm sure very inspiring for all of you who might be actually thinking about a career in science, right? Okay, I think I actually saw a hand over there. I don't know if, yeah. Doris can bring you a microphone.
One simple question. Thank you very much. Well, me as an engineer, I'm really fascinated about the picture of a machine. Any machine needs maintenance. Now we have learned about the life cycle of mRNA. But what is known about the maintenance cycle, or when one of these players, this exomes or something like this, don't work anymore? Is then a problem, or is it known what the cell does?
So first of all, the proteins, the machines also turn over. So these proteins are also going to turn over, and they're going to be replenished. If there is a problem with the machine, it depends where the problem is. So if there is a serious problem, it's going to be lethal. If there is a minor problem, it's going to end up in disease. So for example, in one case, there is a single mutation in one component of this machine. And what it does, we understand now, it doesn't impair, it just decreases the binding of another protein. And what happens with this is a terrible disease called pontocerebellar hypoplasia. And so in this case, if you have a mutation in one of these components, now if it's a really bad mutation, it's going to be lethal. This is an essential machine. If it's a mutation that lets you get by, that the machine get by, is going to have severe effects. So this is there is a mutation. In terms of maintenance, I don't know what you mean, whether if it's correctly done, it's going to be degraded and it's going to be made again. Some of these components are being, of all the adapter proteins that I showed you are going to be made, particular times when they are needed. So it's I don't know if I can push the analogy so much as to the maintenance more than that. Peter.
What a wonderful lecture, Elena. Thank you so much, and wonderful work. So you mentioned this is all messenger RNA, and of course there's lots of other RNA in the cell, all this ribosomal RNA. Yep. There must be quality control there as well.
Oh yeah, all the time.
So other types of exosomes?
Well, actually the first movie was an exosome working on a ribosome. And so in that case, it's not so much quality control. In that case, it's processing. So it's going to mature the RNA in this particle. But there is quality control all the time, particularly in the cytoplasm. So in the cytoplasm, what happens is that one of the adapters of the exosome, which is actually the major adapter, the only adapter in the cytoplasm, recognizes the ribosomes. And the question is, how does it target the right ribosomes? It's obviously a ribosome where translation has to happen, and you have to degrade the messenger RNA. And so we are trying to dissect in that case how this other factor proteins, it's called the ski complex, then can bridge the ribosome to the exosome. And again, we see a lot of sensors and a lot of building plates. What it seems to be is that this machinery is inherently inhibited, and you have to remove which you may want to do. Because it's like a very dangerous thing to have around, and you have to remove these lids at a specific time, and we don't understand yet how this is done in all cases.
Hey, it was a really fantastic lecture, particularly also at the end about the protein-mediated RNA packing. I was wondering, in terms of, RNA can be mRNA can be quite different. We have bigger proteins. I'm from Matthias Mann's lab, so we do a lot of protein stuff. They can be very different from each other. So I was wondering how this protein-mediated RNA packing defines actually the localization, for example, or the lifetime, or how different this looks like, because you draw the comparison to origami. So how different do these protein-mediated packing looks like?
We don't know. It's a huge, unknown. Yeah. So and I think there is not going to be there is not going to be a simple answer. Okay. So first of all, it's unclear. Is the same message going to have the same origami structure, Or is it what is important that it has an origami structure? How does the origami structure change when you go to the cytoplasm? Now, when the ribosomes come through and they go through the message, they're going to unfold it, okay? But what happens just before? And what happens, there are messages that cells have to keep untranslated for a long time. The messenger RNA neurons, they have to travel a long time before they are translated at the end of the synapse. And my bet, or mRNAs in early development, and my bet would be that these ones are going to have much more packaging possibilities than mRNAs that you need to translate immediately as you get out to the cytoplasm. But this is just pure speculation. It's a wonderful thing. You should get into it. You should do it.
Thank you. Let us take a couple of questions from the online audience. The first question is, do you see a similar mechanism in bacteria?
Interesting. So it's a question I've been interested about. Short answer, I don't know. But it turns out Marcela in the lab, the student in the lab, has been purifying messenger RNAs from the human cytoplasm, from the cytoplasm of human cells. What she finds there as a protein is a protein that we know has similarity to a protein in bacteria that is turned on, that reacts in stress conditions. And so I think it's going to be fascinating to see whether there is some kind of packaging is kept in the cytoplasm, whether maybe there is some similarities to what happens in bacteria in some kind of stress conditions. Short answer, we don't know. There's a lot again there to think about.
Very, very interesting. Another question we have in the online, from the online audience. I was wondering what determines the folding in the mRNA, which helps in its quality control. Is this machinery uniform across all tissues?
Probably not. Yeah. But again, we do not know. I would love to think, that as the messenger RNA, I don't know what Patrick thinks about this, or Matthias, and as the messenger RNA emerges from the RNA call 2, it's going to have a certain sequence. So of course, it's going to have a certain preference for folding in a certain way. And some mRNAs are going to have, maybe again, the neuronal mRNAs are going to have more potential for these interactions, and perhaps there is more homogeneity or something in this type of folding than others. I don't know. Is this what Matthias is this a question or a comment? No. Or
if you allow me, I would ask a question.
Sorry. I'm not sure. Is it an easy question?
Very easy. I want to sort of take all of us back to your slide, with a messy office, the desperate guy in the messy office, where there are lots of papers around him. And the question is, which of the papers are the ones that should end up in the shredder? What are the faded papers, maybe, the old ones?
Yeah.
And how are they being recognized by this machine? And, what are the, let's take, Donald Trump's home, before it was investigated. The classified documents that shouldn't be there and that you should be putting into the shredder because they're faulty or dangerous. Okay. So how does the cell recognize
I think I answered the first part. I don't think I'm going to answer the Donald Trump office part. I can speculate. My speculation would be, if it's an important paper, this has made it already to a kind of different part of the desk. Yeah, as in the cell, if the mRNA is perfectly fine, it's going to be taken over by the transport machinery, which is going to, so the exosome or this degradation machinery is perhaps in these cases, they just don't have the window of opportunity to act on. It's the ones that are there that, are not taken over to the next step that I think have a more likely chance then to go. But, then I'm sure there are cases where you can target AU-rich elements and this kind of cytokines and this kind of mRNAs. but for the office messy, perhaps there is some order in the disorder.
Right. We have another question in the auditorium.
Thank you so much for the talk. I just wanted to know how the research which is happening now, or what kind of questions which can be asked next in this research would actually be able to, we can use it in human everyday life.
Sorry, again, which questions are asked that we can?
Yeah, what kind of questions can be asked on this research where we can use the research in human everyday life?
You know, I would say that, you know, if you look at CRISPR, would you have guessed that by studying a phage infection in yogurt, you would have had such a huge impact? No. So I'm not sure, for me, I'm not sure that's, you know, I'm not sure that going after this question is what is going to get to the big revolutions. I would say, you know, to me it's more like, you know, perhaps, you know, to just pay attention to the unexpected, you know, the unexpected bend on a gel, the unexpected DNA sequence, and just try and understand what it does, and then harness it, then think about it, how it can be done, how it can be used. So at least this is, but you know, I am very molecular, I'm very mechanistic, yeah? So this is, you know, the process we use in the lab. So we don't come from this kind of angle.
Elena, can you maybe say a couple of words about the therapeutic or medical kind of implications of your research?
Again, we don't work on vaccines, but I do hope that if we understand how this octopus tentacles work, that perhaps there could be small molecules that could do something similar. In fact, there are small molecules, you know, this molecule called spermidine, that biochemically, you know, if you really push it, you know, tries to do what the octopus does, but it doesn't do it as well. And so I do think there is scope to try and have small molecules. It's not going to be my work, or the work of my lab who's sitting there. You know, we are going to try and understand the mechanism, but I do think there is scope to find molecules that will help package it. Of course, we will have to understand what's happening in the cytoplasm, because this is where the action takes place for these vaccines.
Okay, I'm looking out here. Yeah. Okay.
Yeah, maybe just to follow up on that question. I mean, I believe that BioNTech were originally working on cancer therapeutics, and then they got distracted by the vaccine.
Yes.
And it was very interesting. Both you and Patrick were talking about the revolutionary new discoveries around cancer therapeutics, and you seem to really think that this is a really significant breakthrough, and it'd be wonderful if you could talk a little bit more about that and this idea of kind of personalized, targeted tumor treatment through mRNA.
So this is not my work. This is very different from what I'm doing. So I think it's a fantastic opportunity for the future, which, you know, BioNTech, Moderna, all these companies are going to work on. And, you know, it's a different level of biology that we are working on. But I do hope, as I said, that what we find at the basic, you know, at the basic fundamental biology and chemistry of the process is going to help, you know, BioNTech. And so, you know, a major problem is going to be how do we extend the lifetime of these mRNAs in these vaccines, yeah? And so how do we interfere with the degradation machineries that are going to access it? So I think this is going to be, you know, a problem that can be, you know, they're going to address it, you know, in terms of, you know, industry level and vaccines and the therapeutics. For us, what we're interested in is the mechanisms at the chemical level, but hopefully they're going to be able to exploit it. I think it's a very exciting future. I think mRNA rules the world. The octopus rules the world.
Thank you.
Elena, quick question. So AI seems to be extremely important for the movies that you were showing.
Yes.
And what I'm curious about is how many reiterations and how much work and effort and time goes into the AI integration of the movie? So how long does it actually take until you get to a movie where you feel confident, and how much computing power are we talking about?
So this you would have to ask Janet, because she is the molecular, is the animation lab. I can tell you it takes months, and it takes months of back and forth. And more than the computing power, it takes months of back and forth, because she comes usually with good questions. Shall I put this molecule here, or do you think it's this other part of the molecule? And then you realize, I don't know, I have to do the experiment. And so this is, I would say, is more, it's not so much the movie per se. This is not molecular dynamic simulation. Let me be clear. This is a molecular animation that takes the snapshots and tries and put them together in a way where I give talk also to scientific audience. It just gives the idea of what is a good question to ask next. What is it that we don't understand next? So for this, it took months, but I'm not sure it's Janet who took months. It probably took months for us to the iteration of, what to put in the movies.
Thank you so much. Actually, my question also comes to this process you just describing, and it's I'm a science communication scholar, and I really like the fact that you are telling us that trying to communicate your science helps you deepen the understanding.
Yes.
So my question would be whether events like this, where you do public engagement, whether that's also helping you deepen your understanding of the processes, helping you finding new questions.
I had the movies, I show the movies at scientific audiences too. I think, first of all, science, it's hugely important. I make these movies also for scientific audiences. And the problem is that these machines, I love atoms. I love them, yeah? And I love to look at all these atoms coming together. But it's thousands. If you show all these details, also for myself, I don't understand where to look. I don't understand what's going on. So you really need to abstract, and try and see what's the important part, that you need to communicate. And so I do think that this is important. It's important for the public, of course. It's also important for the scientists, because it gets quite complicated, and if people are not working on exactly the same topic. So I think it's hugely important, and I think it's important, the process of trying to to get to the core of the problem, to get to and try and represent it in a way that gives the idea of what you're thinking. You may be wrong. As I said, the last movie I showed you, I know there are two parts at least that are wrong, because the molecules are not going to magically fly in. But at least it gives an idea of the packaging. So hugely important, I think.
Okay, let us take a couple of questions now from our online viewers. We have a bunch of questions about how quick, about the speed of the exosome degradation, so to say. So how quick is that, and is it always at the same speed?
I don't know the answer to that. Simply because we with the tools we have in the lab, in our lab, looking at bulk degradation. Essentially, if you put an RNA together with the exosome, you see the band of the RNA incubated with the exosome. The band is gone. We don't even see the product. Yeah, it's processing. So, but apart from this, I don't think anybody has tried to actually measure the speed. I think it's going to be a difficult thing to do. I would imagine, so this machinery degrades RNA independently of the sequence of the RNA, so it can degrade whatever sequence. I would imagine if you have a very structured RNA, it's going to take longer. We have tried to do simulation with the late Klaus Schulten, but at the time, the computing power, I think, was not sufficient to do real simulations for such a large thing. So we haven't tried to do it, and I don't know anybody in the field who has looked at it, at this problem, at how fast it is. But for sure, what I showed you in the movie at the end, as I said, was misleading, because that would be very fast at that point when it gets to the cutting.
And we had another question whether actually RNA folding is necessary to be degraded.
RNA unfolding is necessary for the degradation, because for the feeder, as you know, The origami is not going to go through the feeder. Yeah, you're going to have to, disassemble it in a sheet and unfold it to go through. The actual in the feeder part of the exosome is actually a very narrow entry pore, and so the RNA has to be single-stranded to go through. So you'll have to unfold it. And in fact, this, energy-consuming hands that put the RNA through, these ATPases are expected to unwind nucleic acid so that it can go through this channel and then be degraded. So it's actually the unfolding that's important for degradation.
Thank you. I think, we have a question here.
Yes. I would be very pleased if you could give me or us an imagination about how the mechanical processes we see in this video actually take place. Is it known? Because what I have learned from you today is that in such an exosome there has to be an oxygen at the correct position. There have to be other players which select the mRNA and some magical way feed it in. But in the end, I guess also on this scale, basic mechanical rules are necessary. So there are forces, maybe even torques, which act. Is it known, or give us, please, an idea how do you inspect a theory like from the gentleman regarding the AI?
So first of all, the first thing, the hand, the blue molecule, it's a helicase. And so what it does, it takes an RNA and using ATP, so there is one round of ATP hydrolysis for each nucleotide of translocation. And what we think it's going to push it through. Now, if you give, and that's one aspect, then you have, at the bottom, you have the ribonucleotide, you have the cutting, okay? And so what happens, let's say you don't have the hand, biochemically you give you feed an RNA. And what happens is if you have an RNA, so where is it? So this is the feeder, this is these are the 9 proteins, and this is the cutting. So if you have an RNA that can only get from here to here, it's not going to be degraded. If you get an RNA that gets from here to here, what's happening is that on this side, there are very strong binding sites that what we think are doing are pulling the RNA through. It's not a yes or no, it's not black or white, it's a gradient. So essentially, at some point, it has to reach a point where there are a series of very strong binding sites that probably pull the RNA through. Now, how these sites are coordinated with the energy, with the ATP, with the ATP energy above here is not known.
Yes, the microphone is coming.
Thank you, Elena, for this great lecture. I have a peculiar question. How does a living cell, prevent natural messenger RNA, which is to be translated, that it's not packaged? So, because if it would be packaged, it would not be translated. It would not get into the ribosome and would not be translated. So is that known?
Partially. So what I showed is the packaging in the nucleus in yeast. Okay, so this is going to have to go through the nuclear pore complex. So what we think at the moment is that it's going to still be packaged, more or less, until it gets to the cytoplasmic side of the pore. Now, the octopus is going to stay in the nucleus. Okay, so there is going to be an exchange there. But from the purifications in human cells, what Marcella sees is that there are still proteins there, not all proteins. Some HNRNP proteins are there. So it may be, there is going to be an exchange. So at least for the mRNAs that are going to be translated immediately, maybe it's not going to be so packaged. But there are still proteins there. What happens during the first round of translation? I don't know, because the ribosome is going to go through and kick them off.
A lot to do.
This is another question where I don't think there's an answer, but just to hear your opinion. So you've spoken about degrading things that are defective. You've spoken about the machinery being used to degrade things which are not correctly made or heated or are damaged. But in fact, perfectly good, normal RNAs turn over at a certain rate. And the first thing is it exactly the same machinery which does those two things? And second is, what are your ideas, if it is the same machinery, of how you transit from being normal to recognized as abnormal?
So this is going to happen in the cytoplasm. You know, the turnover, the messenger RNAs, it's obviously connected to translation. And I don't know what Matthias thinks about it, but you know, there are features on the mRNA, like the poly-A that seems like an inbuilt timer in the lifetime of the messenger RNA. But we don't understand exactly what that means. The codon sequence also has, you know, anything to do with the, you know, it's going to have to do with the, you know, with the ribosome engagements and the proteins that are on it.
Right. That's good for messenger RNA?
Yeah, for other RNAs. Oh, like, you know, excess of tRNAs in the nucleus, they are degraded. And that goes back to the question from Matthias. You know, it's the same, in that case, you know, or, you know, whatever, defective tRNAs, you know, the tram complex puts a poly-A on it, and then the exosome degrades. But there are other machinery, I mean, I'm very exosome-centric here, but there are other degradation machineries in the cell.
Thank you. I'm slowly looking at my watch as well. So we'll need to wrap up soon. Maybe one more question from the audience here, and we take
so thanks for the talk. So you mentioned you're using AlphaFold into your research. So I'm curious, what are your thoughts on incorporating this type of softwares into basic research?
I love it. I just love it, don't you? I love it. It's like, it's been a game changer, yeah, because it's, you know, of course, you know, it's not that it does everything, yeah, but it just gives, you know, it's a great way to screen computationally for possible interactions. Of course, then you're going to have to test them biochemically. Is it to, I don't know in the lab, I don't know what my lab thinks, but it's great to generate hypotheses that then you can test. You know, if you know, use it if the prediction is solid for a certain type of interactions in a pathway, it just immediately gives you the idea of what to get. The other thing is, you know, before we could get just the very strong interactions, but there are lots of weak regulatory interactions. It would be a pain to go biochemically. So I think it just speeds up what we do, so we can concentrate on the really important questions. And the other ones, you know, it's going to be it's a fantastic tool. Sorry, I'm completely in love with AlphaFold. No, with the possibilities. And there are going to be more AI type of tools that are going to change the way we do things. So I think it's a fantastic time to go into science.
Thank you for sharing such an excitement. We feel with you. And I guess as a closing question, we have a very, very nice question from the online viewers, which is, what is the next big question you look forward to answering in the next 10 years?
What, more than 10 years, no, Patrick? I would love to understand how these mRNAs in early development, or these neuronal mRNAs work. That I would love to understand. How do they function? How do they get there? I would like to see an mRNA from birth to death, atomically, with AlphaFold, which cannot do RNA, by the way, so we're still in business.
Well, thank you so much for the insightful comments, and of course, thank you very much for your questions in the audience as well. Thank you so much for being with us here in the auditorium in this wonderful museum. And of course, online as well. Thank you so much for the Potosaki Foundation for supporting the event. We would encourage you to visit the EMBL website for the recording of previous lectures and other resources about very exciting topics in life sciences research that we are currently working on. And we hope to see you again at one of our upcoming events. Take care and goodbye.